Greetings, esteemed participants, and welcome to a momentous occasion celebrating the marvels of genetics and the global significance of DNA. Today, we gather to commemorate International DNA Day, a tribute to the monumental discovery of the double helix structure of DNA by James Watson and Francis Crick in 1953, as well as the remarkable culmination of the Human Genome Project in 2003. We are privileged to host a webinar that promises to illuminate the profound impact of DNA research across borders, cultures, and disciplines. Throughout history, DNA has served the blueprint of life, holding the key to our past, present, and future. As we embark on this journey of exploration, we invite you all to join us in celebrating the enduring legacy of DNA and its far reaching implications of humanity. Today's webinar is powered by Progenics. Progenics is a genomic solutions is a pioneering leader in the field of genetic diagnostics and personalized medicine, dedicated to revolutionizing healthcare through innovative genomic solutions. Leveraging cutting edge technologies and advanced bioinformatics, Progenics offers a comprehensive suite of services aimed at decoding the complexities of the human genome to enable precise diagnosis, targeted treatment, and informed healthcare decision making. Progenics Genomic Solutions offers a diverse portfolio of services tailored to meet the evolving needs of the medical community, spanning genetic testing for hereditary diseases, pharmacogenomics, cancer profiling, and population genetics research. Whether identifying inherited genetic predispositions, guiding medication selection based on individual genetic profiles, or uncovering the molecular drivers of cancer. Progenics remains at the forefront of genomic innovations, driving advancements that have the potential to transform the practice of medicine. Over to my colleague to introduce our today's speaker, Dr. Annie Hasan. Dr. Krishna Prasad. Krishna.
Thank you, Anusha, and very good morning, ma'am. Today, I'm very much privileged to pleasure that we introduce Dr. Annie Hasan, a distinguished researcher and leading figure in the field of human genetics and molecular medicine. Dr. Hasan's impressive career spans 37 years, marked by significant contributions to our understanding of various human pathologies from cancer and diabetes to neurodegenerative diseases and endometriosis. Here, the very much highlights Dr. Hasan's remarkable achievements, extensive in research experience. Research experience included 8 years of working abroad at prestigious institutions like Wellington School of Medicine, Stanford, Solon Catering, and cancer genetics. Her research dwells into diverse areas within human genetics, molecular pathology, focusing on the genetics and molecular basis of various diseases. With over 130 publications in national and international journals, Dr. Hasan actively contributes to the significant community. Her research findings has been presented at numerous conferences and meetings, solidifying her reputation as a leading expert in the field. Furthermore, she actively shares her knowledge through talks at colleges, hospitals, research institutions, and universities. Dedicated mentor and educator to nurturing the next generation of scientists is evident her supervision of numerous PhD students, with 20 successfully completing their degrees under her guidance. She also actively organizes and participates in training programs and workshops, fostering the development of young researchers. So a fellow, she was nominated as a fellow of the National Academy of Sciences in 2010 and currently serves as the president of the Board of Genetic Counseling India. Dr. Hasan's presence at this international DNA webinar is an invaluable opportunity to learn from a true pioneer in the field of human genetics and molecular medicine. Her insights and expertise promise to enrich our understanding of complex world of DNA. A very warm welcome and thank you, ma'am. Thank you for accepting. and over to you.
Thank you, Anusha and Krishna Sai. Krishna, you know, whatever Krishna has said, you all have to take with a pinch of salt because he's one of my trained interns. Okay, so thank you for inviting me here. All of you know the importance of DNA. If DNA was not there, we all wouldn't be sitting here. What I wish to do today is to tell you about the importance of DNA in both health and disease. Every time I ask a student, what is DNA? They tell me deoxyribose nucleic acid. Correct. That's the full form of DNA. But what's the concept of DNA? Why is DNA now just a term which many people, even the lay public, understands? The word DNA is being used a lot in advertisements. Whether it is, so DNA means different things to different people. When Ford cars say that in dependability and integrity is in our DNA, what do they mean? Does it mean that Ford cars have a DNA? No. What they mean is that something which can be passed on without changing is there. Similarly, you see a DNA molecule on the advertisement of Fair and Lovely. Does it mean that the DNA is changing? No. Here the DNA indicates something which is contemporary. There are several, while I was coming in the bus, I always used to see something called genetic homeopathy. And I would say, what is that? What they really mean to say is that we are doing personalized treatment. Okay, so DNA means different things to different people. But DNA, as we in science, in genetics, in genomics, what we understand is that it is the basis of our health and disease. Sai, Krishna Sai, did you get my?
Yes, ma'am. It's a 1 minute, ma'am. Just 1 minute. We'll get
Yeah, okay. So when is DNA synonymous to a gene? DNA is synonymous to variation. DNA is synonymous to evolution. So DNA has various implications, and today, which is given as the DNA Day, we all have to think what are its implications for individuals, for our patients, for society at large. What is the role of DNA in keeping us all together and also making us unique? Yeah.
ma'am, I hope you.
Yeah, I can see it. You can go ahead with the next slide. Okay. So in science, DNA is that molecule which is very important for an organism to develop, function, and reproduce. It is present in all forms of life, from microbes to plants to humans. And every eukaryotic cell has the DNA in its nucleus. One thing which is very important is the universal genetic code. That DNA carries instructions, and these instructions can be followed by everybody, which doesn't happen even in a lab, correct? When we want a technician to follow instructions, we have to tell in a different style. When we are conveying it to a patient, we have to say it in a different way. But one thing very important is that the genetic code is universal. And that is the reason why there's so much advancement and there's a whole field of biotechnology. where the DNA code, because it is universal, can be used for various purposes. Next slide. Can I change the slides? Yeah. So where is the DNA located? As I said, it is in all the cells of our body. In eukaryotes, it is in the chromosomes along with histones, and changes in the DNA are the changes in the gene, which we see as variations. Next slide. So now, since DNA is the basis of genetics and inheritance, we know that it is the DNA which is passed on from cell to cell and from a parent organism to a daughter organism. So as a result, information is carried from cell to cell and individual to its offsprings, more or less unchanged. Next slide. Yeah, so the parents give the DNA through the egg and sperm, and the single cell zygote then divides the cells, and each cell has the same DNA. So from a 2-cell stage to a 8-cell morula to a blastocyst to a baby, we find that individual cells will have the same DNA. Next slide. So all the cells of the body, whether it's the muscle cell or the epithelial cells or the neuronal cells or the sperm cells, all have the DNA, but the sperm cell and the red blood cells are unique. Red blood cells do not have a nucleus, so they don't have DNA, and the sperm cell has only half the DNA, that is haploid. So every cell has diploid DNA. These words, please, as genetic counselors, as geneticists, as lab technicians, remember, when we are talking about isolating DNA, you have to take a cell which has the DNA. That is, you can't take RBCs. If you want to see the genome, diploid genome, you have to take somatic cells. If you want haploid genome, then you have to take the egg or the sperm. So the cell has the nucleus has the chromosomes have the DNA. Stretches of DNA which code for a particular peptide or protein is known as a gene. So the connection between the DNA and the gene is very simple. Gene is a stretch of DNA which codes for a set of amino acids which forms a protein. Next slide. The total genetic material of an organism is known as its genome. Please be clear with terminology. If you are clear with your terminology, you will be able to convey it to your patients, to clinicians, to individuals in the population who want to understand what genetics is. So the total amount of DNA in a single copy of a haploid cell is the genome. Again, in this slide, you can see gene, chromosome, and the genome. Next slide. Now, the history of the genome, most of you are aware of it. It's just a recap that lots of people for a long time, from the 17th century onwards, knew that there was nucleic acids which transferred information. But they did not know what was the structure of these nucleic acids. And it was only in the 1950s that X-ray diffraction technology was there, where molecules were crystallized, and these crystallized molecules were captured and studied by X-ray diffraction. And Rosalind Franklin was the one who was very good at doing these X-ray diffraction studies. She was in France, and her colleague was Wilkins. Similarly, while she was studying the structure of DNA, James Watson and Francis Crick in Cambridge were looking at the DNA structure by another method. They were making models. They knew that there was a sugar, a phosphate, and the four nucleotides. And they were trying to arrange it in different ways because at that time, there was nothing like molecular biology. It was just chemistry and physics. So they had molecular chemicals and their chemical structures, and they were trying to fit it in to see what the structure would be like. So when they got the X-ray diffraction radiographs of Franklin and what they were trying to do with the model, they came up with the double helix in 1953. Next slide. And for this double-stranded structure, which they proposed, they got the Nobel Prize in 1962, where Watson, Crick, and Wilkins got it. Frankly, she couldn't get it because of the fact of two reasons. One, Nobel Prizes are never given posthumously. That is, if a person dies, they're not given. And she died. She had ovarian cancer. She was depressed that her name was not there in the Nature paper, which came about the DNA structure. And she committed suicide. And as a result, she was not alive when the nominations were made. So although her role in uncovering the structure of DNA is known, but she did not get the Nobel Prize. Next slide. Then, despite the fact that the double helix was known, it was Crick again who unraveled the mystery of how the information in the DNA is passed on. And then came the discovery of the universal genetic code. And genetic code is a language like any other language. Like English has alphabet, 27 alphabets, A, B, C, D to Z. The genetic code has a language which is made up of 4 alphabets, A, T, G, and C. And these A, T, G, and C can make only 3-letter words. In the English language, we have got a single letter can be a word. I is a single letter, it's a word which has a meaning. But in the genetic code, we do not have any nucleotides which together can form a large word like unexpected. No, all words in the genetic code have 3 alphabets only. So that's why it's known as the triplet genetic code. Next slide. So here, this is the central dogma of molecular biology, where the DNA becomes the RNA, RNA translates into the protein. Now, if you see, this is how the DNA is there, A, G, T, C, A, whatever, whatever, whatever. But remember, only 3 of these can code for a particular amino acid. So amino acids have a start codon, AUG, which is methionine, and they have 3 stop codons. And anything which has a start codon and a stop codon with different amino acids in the middle is called a peptide or protein, and the DNA from which it comes is known as a gene. After the Human Genome Project, we came to know that we have 20 to 20,000 genes. And this was very disappointing for the human beings because we thought we are really clever and we are so important. We should have, but we have very similar number of genes to a mouse. Next slide, please. Now, this is a picture of my father and my brother. Very similar. So the father has passed on his genetic material to the son, and they have several similarities. But the father is not an exact copy of the son, or the son is not the exact copy of the father. Why? Because the maternal component of the genetic material is also there, and together the variations of the maternal and the paternal DNA makes a unique individual, which is a child. Next slide. So remember, we think of genes when we are in diagnostic companies and all only associated with disease. But remember, genes and DNA are associated with all the traits, whether it is your hairline, the ability to roll your tongue, the shape of your toes and fingers, the shape of your ear, your teeth, all characteristics are linked to the DNA. Now, this is what happens all the time. When a baby is born, the first thing you hear is, oh, the child has the mother's nose and the father's this and this one, that, and whatever. And my uncle always jokingly says that if you want to know how many flaws you have a child. Because people do not want to criticize you on your face, but they criticize you through your child, saying, oh, the child has a thick bulbous nose like the mother. Or they say, oh, the child has very large ears like the father. So they are trying to describe the child, but also point out flaws in the parents. Now, having said that, there are certain things which we see in characteristics or traits which may not be genetic. Like a carpenter's son is also a carpenter and he uses the hammer very often, thereby his biceps are larger than that of the other people who may be doing craftsmanship or are doctors. So they are not using their physical thing all the time. So certain traits come with the environment or lifestyle. Next slide. So all these traits can be caused by a single gene change or they can cause by multiple gene variants. So that's why we call disorders as monogenic and polygenic. And what it really means is that the DNA change is there in a single DNA, single gene, or in multiple DNA fragments in multiple genes. Genes which together contribute to a disease are also known as polygenic, whereas a single one is known as monogenic. Next slide. Polygenic traits, you can see a variation, like the eye color. If you see Caucasian eye color, you'll find gray, green, brown, black, light brown, dark brown. In India, we usually have brown to black. Of course, based on ancestry, you also see light colored eyes which are coming up. Similarly, if you see height, all of us are not tall, all of us are not short, but we have a variation. And height in inches is the norm if you see a bell curve. On the left and the right are the extremes, who are really short or who are really tall. Similarly, so all these are polygenic traits. where a number of DNA variants contribute to a particular phenomenon, whether it is skin color, hair color, eye color, height, intelligence. Next slide. Next slide, please. Yeah. One thing which many of us forget. No, I want that slide back. Many of us forget that despite the fact that a father has various resemblances to the son, which I showed about my father and my brother. Here I'm showing you a picture of me, my father, and my son. Everybody tells my son that he looks like my dad. And who has passed on this similar looking genes? It's me. So the female. So many times what happens, we totally forget the contribution of the parent, where we are not, like baldness. If my son becomes bald, the trait may be coming from my side of the family, although I'm not bald. Okay. Similarly, breast cancer. Breast cancer in a female may be coming from the father's side, despite of the fact that the father doesn't have breast cancer. So please remember this when you are doing genetic counseling, that the contributing parent may not be of the same gender. Okay, for a disorder which is seen more commonly in a gender specific manner. Like breast cancer is more often seen in females than in males. Baldness is seen in males more often than females. Having said that, you can see both in both parent types, but those are different genes. Next slide. So now we realize that genes or DNA play a role both in health and disease. Getting medals, having good muscles, having resistance to various infections, metabolizing the diet we eat properly, or getting knee pain, diabetes, cardiovascular disease, all this is in our DNA. Next slide. Yeah, so now what is the simplest test? Because now we know that the DNA is passed on from generation to generation. We can do a simple pedigree analysis to know how this particular trait has passed on. And for that, all of you here, at least most of you here who are genetic counselors, will know what a pedigree is. Lab geneticists, clinicians may not know this, but it is very important and very cheap to do a good pedigree to understand the genetic inheritance of DNA in a particular family. Based on this, we are also able to give information about how a genetic disorder or a genetic trait is running in a family. Next slide. The autosomal dominant trait. Like I said, height is a polygenic trait. Polygenic in a particular community. But if an individual has a monogenic disorder called achondroplasia, which is a dominant trait, then 50% of the children will get it, irrespective of the other genes which are associated with height. Got it? So here, a monogenic autosomal dominant gene variant plays a major role and overpowers all the gene variants which are there in for height. Now, autosomal dominant traits, this is a very famous Indian pedigree where the father is achondroplasia, autosomal dominant, and multiple children are affected. Now, if you see, theoretically, we say autosomal dominant condition, if a parent is affected, then 50% of the children are affected. But you have to remember that every pregnancy has a 50% chance. Chance has no memory. It doesn't remember that the first child is normal, so second child should get affected, or 50% like that. Every pregnancy has that 50% chance. And in this family, if you see, they have 5 children, all of them are achondroplasia. So it doesn't mean that autosomal dominant traits do not have a recurrence risk of 50%. It just means that for every pregnancy, the risk was same. Next slide. So there are multiple examples of autosomal dominant traits. Familial hypercholesterolemia is very important. Marfan syndrome is important. And a lot of cancer disorders are autosomal dominant. And that is why there is a 50% risk. And if they are carrying the gene and they have a risk, it is up to us to counsel that that DNA variant is there in the family. Next slide. Next, yeah. So this is an example of Lynch syndrome, where we are showing colon cancer, and that cancer again is seen in 4 out of 5 children, and then going down again they are seeing in multiple children, and the age of onset is reducing. The grandfather got colon cancer at 47. The parents got it in the 40s at 35, and then it's gone down to age of onset 14 and 17. Next slide. Yeah. So similarly, when you talk about autosomal recessive, what we mean is that the DNA given from the father and mother both has a problem, and only if those children who get both variant, both alleles which are variant, then the symptoms occur. But if 2 autosomal recessive parents marry, then all their children will have it. And this is seen in this family with albinism. Both parents are albinos and their 4 children are albinos. So you have to see, you cannot say 25% risk just because it's an autosomal recessive disorder. You also have to see that the parents are affected or carriers. One affected parent and one carrier parent has a different risk. Both affected parents have a different risk, and both unaffected parents have different risks. So please, we know that there is only one DNA variant, but the counseling varies based on what we are seeing in the pedigree. Next slide. I'm not reading out the routine things of the autosomal recessive disorders because all it's there in the slide, and if you want, you can. So a lot of things like cystic fibrosis, a lot of inborn errors of metabolism, thalassemia, sickle cell anemia, all these are autosomal recessive disorders. And in this, we can assume if a child is affected that the parents are carriers. Okay. So we are deducing that the DNA is altered in both the parents, and mostly if we feel they are consanguineous, we expect that the same variant is there. Okay, next slide. So if you see X-linked recessive disorders and traits, disorders, it can be Duchenne muscular dystrophy, or it can be a trait like color blindness. Now, if a father is affected in X-linked disorders, then all daughters are obligate carriers, but all the sons are normal. The mother is unaffected, but is a carrier, then 50% of her sons are affected and 50% of daughters are carriers. So you have to cater the counseling based on what you are deducing the DNA variant to be. In this case, we are considering it to be a X-linked gene. That means this gene is on the X chromosome and it is showing its phenotype in recessive form. And the counseling goes accordingly. Next slide. One more place where there is DNA. Apart from the nucleus, DNA is also there in the mitochondria. It is believed that in evolution, there were plasmid or bacterial DNA which got incorporated in the plasmid-like mitochondrial DNA. And mitochondrial DNA, if it has variants, then it has multiple variants. It's not like I was explaining, mother giving one, father giving one. Here, there are multiple copies of the DNA and there is a lot of heterogeneity. But because the maternal DNA is passed on from the mother, in this case, both the males and females are affected, but it is passed on only through the mother. Next slide. Why is it taking so long to change the slides? Okay. Now we are talking about, up till now we talked about disease or trait. Now we are talking about DNA variants which may increase your risk of getting a disease. Like BRCA1, BRCA2 DNA variants cause increase your risk of getting breast ovarian cancer. They do not cause the cancer. They increase your risk. Similarly, ApoE gene E4 allele causes Alzheimer's disease. So it's increasing the risk. Similarly, here we are talking about single gene variants which are increasing risk, but there are sometimes a panel of gene variants which increase risk, like for cardiac disease, for diabetes, for chronic kidney disease, for asthma, for osteoporosis. You know, there are multiple genes and variants in all of them which are harmful, made to give additional effect of increasing the risk of an individual. Next slide. Based on multiple disease risks, we have got different tests. And this is a very good slide which tells you that 1985 was the first time that DNA diagnostics started. And it was in 1983 that the PCR technique by Mullins was developed. And it was only in 2003 that the Human Genome Project was completed and we came to know about the whole genome. Then by 2014 to 16, DNA testing came into the clinic, which was routine clinic. Okay, next slide. So, DNA diagnostics are also known as molecular testing. Molecular testing, the basis of it is isolating DNA or RNA. Sometimes you can isolate RNA, do a reverse transcription, get the cDNA and do the PCR. But most of them are DNA and PCR based. Even today's NGS starts with PCR based library preparation. So please don't trivialize, but understand DNA testing starts with DNA isolation or RNA isolation, which is converted to DNA, and PCR based amplification. Next slide. So once you have the DNA, what do we look for? You can look for single variants, you can look for single genes, you can look for multiple genes, panels, clinical exome, whole gene. So once you have the DNA, you can do any of this. Now, here we are talking about a single gene variant, MTHFR. MTHFR is the gene. The single variant we are looking at is at the 677th position of the MTHFR gene, a cytosine replaced by thymine. And we can look at it by doing RFLP analysis on the upper side where you see the gel, and you can see the Sanger sequencing below where you can look at the sequence analysis, the same wild type, heterozygous and homozygous variants. Okay, so here you are looking at a small targeted variant. Next slide. Why is this MTHFR important? Because it causes a lot of neural tube defects, it causes young onset stroke, cardiovascular disease, and so on. Now we are looking at the gene. For in ambiguous genitalia, we are trying to see if SRY is present or absent. So you are doing a PCR. Presence of SRY, you see it as a band. Absence of it, you see it as a nothing. So if you see, males will have a SRY band, females will not have it. So an individual with ambiguous genitalia, if they have SRY positive, then you can call it male and do all the other relevant surgical interventions, hormonal therapies for them to get the correct gender of their choice. So remember, now we have genders, we put it as different and sex as different. So this is the genic sex, where the SRY gene or the DNA changes in the SRY, presence or absence decides the male or female. Next slide. Next slide. Yeah. Now, DNA, where we looked at a single gene, where we are looking at large deletions of a single gene, we use another technique called MLPA. But what we are doing is the same thing. We are looking at DNA. It's a molecular technique. So molecular techniques can look at small single variants like we saw in Sanger and by RFLP, and MLPA helps us look at large changes. Next slide. Who's talking? Can you ask them to? Oh, soft. Yeah. Now, if you are looking at multiple molecular markers, we are looking in Lynch syndrome, we are looking at all the MMR genes. By protein, we are looking by protein expression is looked by immunohistochemistry. Then we are looking at multiple by multiplex PCR. We are looking at single BRAF variation by a single gene and sequence analysis. So you see, molecular markers can be. What we are doing is we are looking at the change in the DNA only. The change in DNA is reflected in the protein, which we are looking at by immunohistochemistry. Now, if you see large numbers, if you want to look at, we are doing a microarray panel. So we are looking at a tissue sample of cancer, where by immunohistochemistry, different things you have looked at, and then we are looking at a larger number of these genes by the oncoprint panel. We are looking at genes, and these genes may help us target and do the right therapy. So now we have realized that just by histopathology, you cannot define breast cancer, because breast cancer molecular variations give it a lot of heterogeneity, and that decides on a good treatment response. So next slide. What's the matter? The slide is not changing?
Ma'am, no, ma'am. It's changed. It takes some time to change.
because it's coming nearly to 12:30. That's why I'm worried.
Ma'am, the slide is changed. Can you?
No, it hasn't changed for me. No. Yeah. So the variants we saw there in the somatic breast cancer panel. No. Go back. Now, based on that variant, you give targeted therapy in cancer. But targeted therapy targets the changes in the cancer, but it is that therapy still needs to go through the metabolism of an individual. So the individual's metabolism may be different. And that is why we have to also do pharmacogenomics in these people. So targeted therapy targets the DNA changes in the cancer tissue, but the drug which is given is still being given by IV or orally. So it still needs to be absorbed, metabolized, act, and excreted. So the genes which are responsible for all these steps are the genes which we call, which need to be tested by pharmacogenomics to give a personalized therapy. Next slide. So the patient group has to be again divided or based on the drug you are giving, whether they are good responders, they will get adverse reactions, they may benefit but have some toxicity, or will not benefit at all. So that comes as companion diagnostics. That is, you know which drug you are giving, then you test whether that drug is absolutely fine for that patient or not. And that comes as companion diagnostics. Now, next slide. Now, coming to another very interesting part of DNA. We started by saying the DNA in all the cells of the body is same. We are right. But the DNA in all the cells is not expressed similarly. Like DNA or the gene for gastric juices does not get secreted from the skin. The melanin which is present in the skin should not get secreted in the stomach lining. So the from cell to cell. And this altered DNA expression is because of mostly methylation or acetylation of the DNA. So to study this methylation, the technique used is totally different, where we try to do methylation specific PCR. And many diseases, there are about 200 different genes in our body which are differentially methylated. And we look at that. And this methylation pattern may vary in disease and non-disease tissue. Like cancer may have a totally different methylation pattern of certain genes as compared to normal tissue of the same organ. Angelman syndrome. So you do methylation assays. Next slide. Similar symptoms can occur because of different gene DNA being affected. We got two male children below the age of 1 year, which came with very similar symptoms, where they were getting several frequent episodes of blood-stained stools. Both had a history of one SIB expiring in the neonatal period. We did clinical exome, and what did we find? In one child, there was a IL-10R alpha exonic mutation. In another, we had IL-10R beta. But both were showing same symptoms. Why? Because we know that IL alpha and beta together form a quadruplet complex which acts on the STAT receptors to go ahead and cause inflammatory bowel disease. So you see why doing exome was important? To understand which DNA was affected. Now, why is, although the treatment will be symptomatic, what is important is that for doing prenatal testing for the next children, if we did not do the DNA testing, we would not be able to help the family. Okay, next slide. Now coming to the wellness part. When you look at athletes, you know, people, football players, Maradona, basketball players, they all have a unique physique which suits them and gives them an advantage over the others to perform at a high level in their sporting activity. Now, genomic profiling tells us about the suitability of their endurance, strength, speed, vulnerability to injury, and their nutritional requirement. All this is there in their genes. Next slide. What type of an athlete are you? Do you want to know that? What is your body type? What is the thing you have to do in type of exercise, type of diet to get the maximum potential of your genes? It is very well known that you may be born with the best genes, but if you don't utilize it to the full potential, there's no point. Like a child is born with very good genes for height, but you deprive the child and the child is malnutrited, that he does not get enough protein at the right time during the growing age, then the child will not get the full potential of height. Similarly, intelligence. The child is intelligent, but is not, he or she doesn't get the opportunity to utilize their full potential because they're not given the right environment, they're not given the right education, not meeting the right peers, and that is why they're not able to get their full potential. So similarly, what type of athlete are you? Are you motivated to do the exercise? That's the BDNF gene. Are you prone to soft tissue injury? COL1A1 gene. Are you going to build muscle mass? You know, you can do a lot of strength building exercises, but you may not get muscle building genes. Another is, are you going to get your heart rate recovery quickly, or are you going to get arrhythmias, cardiac sudden death? Will you be able to burn your fat to get enough energy? So there are several genes which help you decide to utilize your full potential. Next slide. Yeah. So DNA plays a major role in your health and disease. But who can help you understand what these are, what the DNA changes are, whether these DNA changes are significant, not significant? DNA changes are there to make us unique, but can they be utilized to make us exceptional? What all this can be utilized and done with the help of genetic counselors. They may help you understand your DNA for your own betterment, to prevent you from disease and introduce wellness and a lifestyle which will benefit you. Thank you.
Thank you, ma'am. Thank you for an outstanding presentation. Your use
And
of
that was
infographics, illustrations, and pictures brought the topic to life in such a captivating way. Each visual aid enhanced our understanding and making us the complex ideas easy to grasp. And your dedication to simplify the subject while keeping it engaging, that was truly made the session memorable, ma'am. Thank you so much for sharing your
thank you.
expertise and for making the world of DNA so accessible for us. We are so fortunate to have you in the field, ma'am. Thank you so much.
Are there going to be any questions?
Yes, ma'am. Yeah, we have a Q&A
Okay.
session. So if please raise the hands, then we'll. Yeah.
I can see one raised hand.
Yeah, ma'am. Shubham, you can please unmute and you can ask the question.
Okay. Hmm. Is there some way you have to unmute them?
Yes, ma'am. We have unmuted. Soham, you can also can unmute yourself and you can ask the question.
Thank you, Krishna. Thank you so much. Good morning, everyone. Thank you so much, ma'am, for this wonderful talk. It's always a pleasure for us to see you, virtual or physical, whatever manner. But it's a
Thank
long
you.
time we have seen you. And we are eagerly waiting for that BGCI to meet you, ma'am. And I think I have shared this talk with some teachers because, ma'am, we are in a try to start the course here. It's a long run, I know, but still we are in trying. And it's a wonderful
Okay.
talk, ma'am. And I believe we need some more talks regarding this, not only about DNA, about the new testing technologies which are coming forward. We need to know that in a details basis. So if you could have arranged something like that, like online mode, so that we can arrange, we can join everyone from the each side of India. That would be a very good help for us.
Thank you, Soham. Thank you for that. Yes, and I think it's a good idea that we all remember and mark our calendars for 5th, 6th, 7th of July 2024 for the Bangalore Board of Genetic Counseling Conference. Yes, because various people had asked me, I have put in 20 talks of mine of 20 minutes each in Udemy online for people to hear, because I thought, 40 minutes, 1 hour is too long in this age of social media where you all don't even have patience to look at a 3-minute video. So, but that's why I consigned it to 20 minutes and did 20 minutes talks. It is online on Udemy, but Udemy has put on its own charge. I don't know why. But if you all want to go through it, no issues. You are most welcome. Yes, Shobham,
Yes,
and
yes,
Aishwarya
ma'am.
also has some.
We have shared your course details with our upcoming students and fellow GCs, but Udemy have put a charge around 6,000 rupees for that.
I don't know.
So
Yeah, I don't know why they have done it. What you could do is write to that, and I will pass it on to the administrator to see if we can do something.
surely, ma'am, surely we'll do that.
To,
Thank you, ma'am.
to
Thank you so much.
reduce it for some of. Yeah,
Definitely.
tell me. Aishwarya wanted something. Aishwarya.
Aishwarya can speak now. We are unable to hear you, Aishwarya. Can you please speak away?
Anybody else? Okay. Krishna, do you think you could ask Aishwarya what it is? Because we can't hear her.
Yes, ma'am. I'm communicating through WhatsApp. 1 minute. If possible, Aishwarya, can you please write in the chat box? Then we can.
Yeah. So I'm really thankful to Dr. Prasad who organized this on DNA Day, and there are various new people I see which I'm not familiar with. Amrit Kalar is new. Himanshu is new. Amrit, where are you from? Can we know this so that we know what they are interested in?
Ma'am, while they're coming back, this is Prasad here. Sorry, I could not connect to you yesterday. Thank you very
No
much,
worries,
ma'am.
sir.
Thank you very much, ma'am. You have agreed within very short notice period of time. And this is very much enlightening. I think though so many aspiring genetic counselors and experienced genetic counselors, many of people I can see in this present talk, they have joined in this webinar. Very good opportunity, ma'am. Still at this age, you're very much updated and giving very good new insights to the people. And very much thankful, ma'am.
Thank you for having me.
Ma'am.
Yeah, in the chat box, I see some questions.
Yes, ma'am. We have a couple of questions. Aishwarya is asking that,
Yeah.
ma'am, actually my sister, she is of 1 year age and she is suffering from tRNA and my DNA mutations. So will there be a change that she can be cured?
See, Aishwarya, I didn't touch upon treatment because we thought first let's understand the DNA. We need to understand what the disorder is. Sometimes, like hematological disorders, they are very easy to treat, where especially with CRISPR-Cas9 technology coming up, sickle cell and all, they are now FDA approved, both sickle and thal. In your sister's case, we need to understand what it is, what mutation is it, like for Duchenne muscular dystrophy. By the time we see the symptoms, the DNA of the muscles gets already disintegrated, and there is a hypertrophy, and then correcting that is very difficult. Whereas hematological disorders or systemic disorders, they are more easy to correct. So it will not be fair on my part to tell you that it can be cured without understanding the disease, and I don't even want to disappoint you in saying that it cannot be cured, because at first I need to understand that. And you could connect with me or any of the genetic counselors, give us details, and that will help us explain to you what can be done in the current scenario. Is that okay, Aishwarya? Next is Pavitra. How does epigenetics influence inherited traits, and what implications does this have for genetic counseling? Yeah, epigenetics does, see, polygenic disorders like diabetes, hypertension, wherever there is multiple genes involved, then we say that there are outside lifestyle factors which are changing it. Epigenetics plays a major role in cell-to-cell differentiation. I explained that, correct? So every inherited trait also, if you are just taking BRCA1, for example, if you're saying BRCA1, single variant, other is normal. One is variant, one is normal. If the normal one gets methylated, then it becomes a double hit. So whole of Knudsen hypothesis for hereditary cancers works on epigenetics. So you have an inherited trait, the other is a methylation, making it a double hit, causing the disease. Okay? Yeah.
So,
is there a molecular?
I'm sorry, ma'am. Yeah.
Yeah, tell me.
Somasundaram, ma'am, he's asking some raising the question, so we are unable
Yeah,
to.
he's,
Yeah.
yeah, Dr. Somasundaram is asking, is there any molecular study for IBS? Yes, IBS is there. That in fact, I showed you one family, two families where they had neonatal uh, colitis and something very similar to inflammatory bowel disease. And we look at a lot of this is IL-1 receptors. So what we recommend is if there is IBS, do a exome and look for autoimmune diseases, IL-1s, IL-1 receptors, and if there are variants in that, it can show it. So IBS does show a genetic preponderance, and diseases according to that can be checked. Now, is that okay, Somasundaram? Now this is Gargi.
Gargi, you are unmuted. And ma'am, we have one more question from Eliza Fatma. Does lifestyle play a role in wellness genomics, and how to integrate that in
Yeah.
genetic?
Oh, that's the up and coming profession for genetic counselors, wellness. Uh, See, what happens is when you get a genetic issue, supposing the simplest example is Federer. Federer, you all know, he's a tennis player. His pulse rate always ranged between 38 and 40. Now, when his pulse is 38 and 40 at normal, after his fifth match, also tennis match, it doesn't rise above 70. So he's not tired, he's not breathless, he's not doing anything. That is because of his gene for arrhythmia. Then, added to that, he has trained himself. You know, sports training gives you that. That's the lifestyle. And that gives it. Similarly, ACE gene. ACE gene, DLL is there. That increases your blood pressure. So people who have the DLL, if they are given drugs to reduce blood pressure, then they can climb mountains without feeling breathless or fainting. And this has become very important for army personnel. Army personnel have to go, in our borders, like the Nepal border, the Tibet border, the Himachal border. There, if you send individuals with a DDLA, they will not be able to cope with it. So the genetics will help you decide who can perform better in those high altitudes. So the lifestyle and all changes with your genes. And your genes help you decide on a lifestyle which gives you the baseline wellness. More recently, you must have heard of a film actor who died immediately after doing gym, and he was considered very healthy. Why did that happen? Because of cardiomyopathy. He had a genetic predisposition for cardiomyopathy. And that is why the exercises which he did were inappropriate for his genotype. Okay. Can I look at this thing later? Because I cannot talk and look at reports.
Okay, ma'am. We have a couple of questions from Gargi Kumari.
Yeah.
Ma'am, if
One is
during
Shubham Kamble. 1 second. I'm affected with muscular dystrophy, but type has not been diagnosed. Done DMB, MLPX negative, clinical muscular dystrophy negative as well. And also negative. I'm a genetic counselor too, interested in research. Okay. Okay. See, are you sure it is muscular dystrophy? The question is, was the initial diagnosis correct? Because the exome sequence analyzes only what phenotype you have written. If you have written muscular dystrophy, then they will do only those genes. And the bioinformatics looks only at that. Muscular dystrophy can also be myotonic. No, a muscle biopsy we are not looking at now nowadays. That's why I wanted to know what is your CPK, and we have to look at the CPK. We have to look at the thing, your other clinical phenotype. Is your only your lower limb affected? Is your upper limb affected? Is it affecting your internal organ musculature like swallowing and so on? So I think it's not right for me to make any thing. You first have to give me the information, and I think a webinar is not the place for me to do genetic counseling. You have to agree to that, okay? You have to give me enough information at the right time. And so, Krishna, I think all the people who are interested in the counseling part, you can take them details, make case reports and share, and then I'll be happy to discuss. Is that
Definitely.
okay?
Yes, ma'am. I'll look into that. And we have a couple of questions, ma'am.
Yeah.
Can I?
Yeah.
Yes, ma'am. Ma'am, is during the prenatal testing, the reports comes as normal, and then after the birth, the child is having some disorder. Then, in that case, how we should counsel the parent?
Listen, it depends on which test you have done. Choosing the right test is very important. Doing a prenatal test, which one have you done? Have you done a chromosomal microarray? Have you done a karyotyping? Have you done a molecular test? What have you done? And what is the disorder coming out as? If you have done a karyotype and the chromosome is normal and the child is born with MPS, then you have not done the right test at the right time. The prenatal testing, like if you take blood from me and tell me you don't have diabetes after doing a thyroid test, then you are not doing the right test, correct? For you to tell whether I'm diabetic or not, you should look at my fasting sugar, postprandial sugar, glycated hemoglobin, something like that, to tell me that I'm diabetic or not. So prenatal testing itself doesn't mean anything until you tell me that, okay, I did a prenatal test for karyotyping and still a Down syndrome came. Then that's a worry because the test was not done properly. Where did we fail? Is it a mosaic that was missed or something? So the type of test done and type of disorder identified after birth indicates whether you have chosen the right test or not.
Yes, and we have one more last question. How can genetic counselors effectively communicate complex genetic information to patients with varying levels of scientific literacy and cultural backgrounds?
In fact, scientific literacy needn't be there in your patients. You are supposed to convey it. Now, conveying, it is said that Einstein believed that if you know your subject well, you should be able to explain to a layperson. And if a layperson understands, it's only then we can believe that you know your subject well enough. So, education has nothing to do with communicating to an individual. A person may be a PhD in maths, but may not know biology. Nowadays, it's becoming worse because the other day I got a 14-year-old boy who is doing, he's preparing for his, I don't know, engineering or something. So he's studying in Nairana or some such place. He hasn't even done 10th class biology. So where I assume that he's now in the 11th class preparing, so he's a student, he should know. And I started, and then I asked him, and I realized he has no knowledge of biology. So you start from the very beginning and convey in terminology which they understand. There is no need to translate words. Though you don't have to say DNA is deoxyribose. No. DNA, they will understand the word. Say DNA is this, which is there in your cells, your body has this. Start from where they understand and build on that. It has nothing to do with scientific literacy. It is your scientific strength which you should be able to convey. Yes, somebody else is saying, thank you, madam. I'm a retired scientist, deep from Central Sericulture, Hosur. Yeah. So the gene which I discussed, the MTHFR gene, sir, becomes thermolabile. Okay, if the cytosine is converted to thymine at the 677th position, and at 37 degrees it cannot work, and that is why you get elevated, because it's not able to metabolize, you get homocysteine accumulation. Anything else?
No, ma'am. I hope the questions are done.
Okay,
Thank you, ma'am.
so much.
Yes, ma'am. It's a great pleasure to have you, ma'am, today. Thank you. Thank you for everything.
Thank you. So for the success of the webinar, Sai Krishna, what you have to do is you have to talk to the people who want help, like
Definitely.
the 2 or 3 who talked about disorders. Talk to them, get their information, look at what genetic tests they have done, see what is missing. You can discuss with me, my group, anybody else, and we can talk about if any other further testing is required. Okay?
Definitely, ma'am. Yes, ma'am.
Yeah.
Thank you.
Thank you.
Thank you. Thank you, ma'am.
Bye-bye.
Bye.